Good morning, Year 9, or afternoon, Year 9. Welcome to week 5. This is lesson number 1 of week 5. Hope you're all well and had a good weekend. We'll be continuing on, obviously, for until they announce anything different. So let's get going with week 5. In this lesson, we're going to be looking at how the digestive system works. Now, we've done a lot about the digestive system and enzymes so far. What we're particularly going to be focusing on in today's lesson are the names of the groups of enzymes, what it is that they break down, and what they get broken down into. So it's something for you to think about before we get going. If food travels in the blood, why don't you see your food when you cut yourself? So why isn't it that if you cut yourself or bleeding, why is it that you don't see chunks of your meal in it? Okay, so hopefully we've had a think about that. And hopefully we've got the idea that obviously it is broken down from the food into the kind of basic structures, the monomers or the single units such as glucose, amino acids, those kinds of things. Okay, so what do we need to leave this lesson knowing? We need to know or recall some of the enzymes that are used in digestion. We have mentioned some of them so far. We also need to know a couple of examples of the groups of enzymes. We need to explain why enzymes are needed for digestion, and which enzyme breaks down which food group. And also thinking about how we can test substrates and products in a gut. Okay, so we've spoken a lot about enzymes and what is special about them, how it is they work, what the process is that they actually break things down. So the job of digestive enzymes is to break large insoluble molecules into smaller ones which are soluble, that can then be taken around in our bloodstream. So all the food that you do eat does end up in our bloodstream. It's just that it's been broken down from those big molecules that we eat into much smaller ones. So quick question, what is an enzyme and how does it work? So pause the video, you don't need to write anything down, but pause the video, try and remember what the definition of an enzyme is and how is it that it works. The diagram on the right hand side will hopefully trigger your memories for that. Okay, so hopefully you remember that enzymes are biological catalysts. So they are things that are made in the body, proteins that are made in the body that speed up the rate of the reaction without themselves being changed. How do they work? We use the lock and key theory. So specific shaped enzymes break down specific shaped substrates and they come together from an enzyme substrate complex. Then there's the reactions that take place, and then you end up with your products. Okay, so most of the enzymes that we make loads and loads of different enzymes. They're used for loads of different chemical reactions inside our cells. And that's what most of them do. They just remain in the cell, controlling all of those chemical reactions that take place. But digestive enzymes are different because they're not working inside the cell. They're produced by different glands, and then they get to work outside the cell. And some of the glands, the main glands that are used to make enzymes or secrete enzymes, are the salivary glands and the pancreas. There's also glands in the stomach that also secrete a specific group of enzymes. Okay, so we're going to go through 3 sort of main groups of enzymes. ones that break down carbohydrates, ones that break down protein, and ones that break down fat. And there's certain things that you need to know about these enzymes, which we've put together on this table. So I am going to ask you in your notes to copy this table. If you do have access to a printer, by all means feel free to take a screenshot and print it. But you do need to make sure that it is quite big, or just draw it out in your book, trying to take up as much of the page as possible. So on the left-hand side in the first column, we've got some questions that we need to, or some different things that we need to know about. So why do we need this food group? We've already spoken about that in a previous lesson. An example of a food source that contains this food group, again, something that has come up in previous lessons. Name the enzyme that breaks it down. This will be new. Where is the enzyme made? Again, this will be some new information. What does the food molecule break down into? Okay. Now, there are some keywords at the bottom, but you will see those important words as we go through the information. So what I'd like you to do now is pause this video, get this table in your notes, and then we're going to go through the 3 different ones where you can fill in your table. So can you do that now, please? Okay, so hopefully we should have a table ready to go. We're going to put the information in. Over the next 3 slides, we're going to talk about each food group and what it is that breaks it down. There will be a lot of information, so you need to pick out the information that answers those different points. So we're going to be starting off with digesting carbohydrates. Okay. Now, I'm not going to read out everything that is on the slide, so you might want to pause this at the end and then fill in your table for the first or the second column, which is carbohydrates. We've already spoken about how important they are because they are what provide energy in our body, so we need that energy to do lots and lots of different things. We use if you take a complex carbohydrate, then that is broken down into simple sugars. Okay, and it's those simple sugars that we use in things like respiration. Now, carbohydrates are found in quite a few different food groups. So bread and cereals, both from plants, vegetables, legumes as well, so that's like your beans and stuff like that, pasta, rice, all of those will contain carbohydrates. Now, what is important and will be new information is what's on the right-hand side. It tells you the name of the group of enzymes that break down carbohydrates. Now, there's lots of different types of carbohydrates, so there'll be lots of different types of enzymes that are breaking them down. But they all come under a group of enzymes called carbohydrates. Okay. Now, carbohydrates are enzymes that break down carbohydrates into simple sugars. So that's what we need to really make sure that we have down. Carbohydrates break down carbohydrates, and they're broken down into simple sugars. Now, an example of a carbohydrate is amylase. Okay, now amylase is one that specifically breaks down starch, and it breaks down starch into glucose. So there's one carbohydrate you need to know about, that's amylase, and there's one, the next slide, that breaks down protein that you need to know. So please make sure that you have got amylase in there as well as carbohydrates. So the group of enzymes is carbohydrates. An example is amylase. So you might want to pause the video now, read through the information, and complete that column of the table. Okay, so we're going to move on now And next up, we have got the proteins. So in the previous lesson, we spoke about where we get our protein from. So it can be things like nuts, soya, tofu for vegetarians, but also meat, fish, and eggs. Now, protein is really important because it helps build up new cells, so we can replace cells, we can repair cells, and also we can make things like enzymes. Now, proteins are broken down by a group of enzymes called proteases. So notice that both of them so far end in A's. So if you see an A's in biology, it normally means it's an enzyme. So proteins are broken down by proteases. Now, one example of a protease that you do need to know about is called pepsin. You can see that word in bold in the middle. This is one that is produced in the stomach. So it works best, it has an optimum condition of much lower pHs than in other parts. Now, proteases aren't just made in the stomach. They're also introduced in the small intestine as well, and they won't work at such low pHs. Now, proteases will all break down proteins into amino acids. So amino acids are what build up proteins. That is what proteins are broken down into. So what's really important from this slide is that proteins are broken down by enzymes called proteases, an example being pepsin. And proteases break down proteins into amino acids. So can you pause the video, have a read through, and then make sure that you've got your column on proteins completed on your table. Okay, next up, we have got the fats, or the lipids as they are better known. Fats we've already spoken about, very, very important because they are a source of energy, but also help with thermoinsulation and also help absorb different vitamins. If we didn't have the fats full, we wouldn't be able to absorb. Now, lipids are broken down by a group of enzymes called lipases. So carbohydrates are carbohydrases, proteins are proteases, lipids are broken down by lipases. Now, there isn't a specific one that you need to know about, unlike the carbohydrates, which is amylase, and the protease pepsin. You just need to know that lipids are broken down by lipase, and they produce fatty acids and glycerol. Now, this is the one that most students forget in the exam because there are two things that it is broken down into. It's not as straightforward as the others. So we do need to try and remember that lipids are broken down into fatty acids and glycerol. Okay, so if you would like to pause the video, have a read through the information, and then complete the column on your table. Now we're going to go through just the very, very basics of what should be in your table now. So grab a different colour pen and then tick it off if you have got the information, or fill in any that you are missing. So if we go down the column, starting with carbohydrates, it's our main source of energy. You can find it in things like bread, and pasta. It's also in fruit, rice, anything like that. They're broken down by carbohydrates, such as amylase. Where is it made? It is made in the salivary glands, and it's also made in the pancreas and the small intestines, if you got that from the slide. And what's it broken down into? They're broken down into simple sugars. So an example of a simple sugar is glucose. Right, next up is protein. Why do we need it? For growth and repair. Where do we get it from? Meat, cheese, beans, which are the legumes, eggs, fish, anything like that. What's the enzyme that breaks it down? It's protease. And an example we need to be familiar with is pepsin. Where are they produced? They're produced in the stomach, the pancreas, and small intestines. And what is it broken down into? Amino acids. Okay, the last column, fat, it's for insulation, store of energy or use of energy. Where do we get it from? Cheese, milk, meat. What's it broken down by? It's broken down by lipases, which are produced in the pancreas and small intestine. And what are they broken down into? It's broken down into glycerol and fatty acids. Okay, so we're going to team what you've just done with some drawings to show how it is that it is broken down. So you can see here, this is taken from a revision guide. You can see here we've got some long chain molecules. That is what is being broken down. That would be the substrate. We've got an arrow showing the reaction and the name of the enzyme, the arrow. And then we've got the products on the right-hand side. So what I'd like you to do is just a quick diagram putting down the name of the substrate, the arrow with the enzyme, and then the name of the product. Okay, so hopefully we have got our diagrams drawn and the important parts added onto there. And we should have for number 1 will be carbohydrates. You might have specifically have said starch, that's fine, but it's carbohydrates. And the products will be simple sugars, or you might have put glucose. Either one will be acceptable. Number 2, so that was for number 4. Number 2, the name of the molecule is a protein, which is broken down by protease. It's broken down into amino acids for number 5. Number 3, we've got a lipid that's broken down by lipase, and it's broken down into fatty acids for number 7 and glycerol for number 6. Okay, so something to have a think about, but you don't need to write about. Why can a protease enzyme not break down starch? And what pH do you think the enzymes in the stomach and small intestine work best at? So pause the video, just think of the answer for those 2 questions, but you don't need to write them down because they are things that we've spoken about before. Okay, so hopefully you've had a think about that. So the question, why can a protease enzyme not break down starch? And hopefully you use the word specific in your answer because they have a specific shape. So something about the starch not being able to fit into the protease enzyme or the active site of the protease enzyme. But the question, what pH do you think the enzymes in the stomach and small intestine work best at? So obviously the ones in the stomach are much lower, so perhaps you said 2. Small intestines will be higher, more towards neutral, so we should be going for about a 7. So the last kind of thing, putting everything together that we have spoken about in terms of enzymes, the practical that Miss Corbett went through, the different enzymes there are, how they work. So we've got this question. It says, explain the importance of the digestion of food in terms of the molecules involved and the role of enzymes in the gut. So that's going to be a 6 marker. So we need to explain, so we need to say why for that question. So explain the importance of the digestion of food, so why we need that food and what molecules and enzymes are involved in it. And if you want a little bit of an extra challenge, what would happen if we didn't have these enzymes? And don't just say we would die. Yes, that would be the end result. But why is it that we would meet that fate? So if you could pause the video, have a go at the 6 marker, and then have a think about the extra challenge if you want that extra challenge. Okay, so we're going to go through some different places you might have got marks from. So grab another coloured pen, and we're going to go through the answers and just tick where you have put this in your answer. So you could have said something about the molecules, large insoluble molecules can't get into the blood. So they're broken down into small soluble ones. Sorry, that should say soluble, not insoluble. Soluble molecules, that would be a mark. If you said about enzymes speed up that reaction or speed up that breakdown, that would be a mark. So then you can talk about where it's happening in the gut. So you could talk about how some carbohydrates are broken down in the mouth, that would be a mark. You said about some proteins broken down into the stomach, that would be a mark. You said about all of them being broken down in the intestines, that would be a mark. And then if you said that they're absorbed into the body, that would be a mark. If you said about how carbohydrates break down carbohydrates into simple sugars, that's a mark. If you said about proteases breaking down proteins into amino acids, that would be a mark. And if you said about lipases breaking down lipids or fats into glycerol and fatty acids, that would be a mark, with obviously a maximum of 6 marks. So hopefully we have got 6 or near to 6 marks for that. And that's kind of bringing everything that we've done together over the last few lessons in kind of one go. So the last thing that I'd like you to do, and we're done with writing, so you don't need to write these down. We've got some fill in the gaps that I'd like you to have a go at. So the first one is, where are the following digested? And you've got the 3 groups. 2, what are the following broken down into by digestive enzymes? Again, with the 3 groups. And then the third one, where are most enzymes produced? So pause the video, have a go at answering those in your head, and then we'll go through the answers. Okay, hopefully we've had a go at those. So where are the following digested? Carbohydrates are in the mouth and in the small intestine. Proteins are in the stomach and small intestine. And fats are in the small intestine only. What are the following broken down into? Carbohydrates are broken into simple sugars, proteins into amino acids, and fats into fatty acids and glycerol. And where are most enzymes produced? In the pancreas. And that gets released into the small intestine. So hopefully from these lessons we should be able to name the 3 sort of main groups of enzymes. Hopefully we should be able to give the example of carbohydrates, which is amylase, and the example of the protease, which is pepsin. Hopefully we know how to test for the different substances. That will be bringing in what we did about the food testing, so how you would test for starch, simple sugars with the Benedicts, protein with the Biorect, and the fats with ethanol. Hopefully we can explain why enzymes are needed for digestion, and we can also name which enzyme acts on what and what is produced. And then for the higher levels 7 to 8, suggest how to test for the substrates. We've already said that one. And also evaluating models by discussing its limitation. Now that one again will be more relevant in the next lesson, where we look at some of the adaptations of the digestive system. Okay, so that is it from me. I hope you were able to follow. If you have any questions on any of these lessons, please feel free to email me at sdeaton, which is DEIGHTON, at knoleacademy.org, and I'll get back to you as soon as possible. Take care of yourselves and your family, and you'll be hearing from me soon. Take care. Thank you. Bye-bye.